Hello, we're going to do the next part of our plant hormones and carrot tissue culture lab. And in this one, the tissue will be prepared and placed onto the culture medium. So the first thing I like to do is actually wash the tissue. In this case, they're just carrots that I purchased from a market. So there's nothing really special about them. And I just like to give them a good scrub at every step along the way. Anything that can be done to minimize bacteria or fungi that'll get transferred could be transferred to the medium, those steps should be taken. Next, I always like to wash my hands really well up to the elbow. The sterile part of this procedure will be performed in a laminar flow hood, and so anything that will be introduced to the laminar flow hood should be washed or sterilized. And so I will be placing my hands and some of my forearms into the laminar flow hood, so I like to give them a good wash. Next, I'm going to prepare the tissue, and this is not done in the laminar flow hood. This is out just on an open bench. And so I'm going to take the carrot and I'm going to cut it into a 5 by 5 millimeter by 5 millimeter pieces. First, I'm going to cut off the epidermis. What we really want is the parenchyma tissue that's in the interior. That's what's going to grow into callus for us. And these little blocks are what will eventually be will be cut down farther and placed into culture. And I'm just going to continue that process until I have all the tissue I think I need. So the last thing I'm going to do is pour a 1% sodium hypochlorite solution into the beaker, and that is to surface sterilize all of those tissues. And so this is essentially bleach. So standard household bleach is 5.25% sodium hypochlorite. And if you make a 20% bleach solution, which is about 20 milliliters of bleach, and add 80 milliliters of distilled water to that, then you end up with about a 1% solution of sodium hypochlorite. And this is enough to kill most bacteria, fungal hyphae, or fungal spores that might be adhering to the exterior of the tissue. And you've got to kill those before you place them onto the culture medium, or they will outgrow your plants, your plant tissue. So next we're going to rinse the bleach away from the tissue. The tissue, the plant tissue, has been in the bleach solution at this point for approximately 15 minutes. And I like to put it in there and then put the tissue in the bleach solution, and then I'll wait 5 minutes and check it, and then another 5 minutes. And when the carrot tissue is starting to bleach or turn white, that's when I figure it's ready. So it's been placed into the hood now. And the first thing is I'm unwrapping the instruments I'll need. They were wrapped in aluminum foil and autoclaved, so they should be sterile. I'm using a pair of forceps just to hold the tissue back while I pour off the bleach solution. So I have several Erlenmeyer flasks with sterile distilled water. So it's distilled water that's been autoclaved. And I will just use that to rinse the tissue, rinse the bleach off the tissue. And I will do that. I'll do the rinse 3 times. The waste beaker I'm using right here has a crack in it, and you can see the water seeping out. I didn't realize that at the time. Now the tissue is ready to place onto medium, and you can see I've lit my Bunsen burner back here, so that's burning. And these are just paper towels. It's a good idea to autoclave something absorbent, just in case you need it. I thought I was going to have to blot the tissues onto it, the surface sterilized tissues onto them, but ended up that wasn't necessary. I probably should have used them to absorb that water and bleach solution that's on my working surface, but in the moment it didn't seem very important, so I didn't do it. So my instruments are already sterile from autoclaving, but I give them a quick flame them quickly just to ensure that they are sterile. Some people will have 70% ethanol in the hood with them to dip their instruments. I didn't in this case because I had autoclaved my instruments. So I'm cutting the tissue. I'm using the forceps to hold the pieces of tissue, and using the scalpel to cut them into sections that are approximately 3 millimeters thick. And I will place the tissues onto the culture medium. I'm putting 2 pieces on a plate. You could put 3, you could put 4. I just chose to put 2. And I'm just going to keep placing tissues. So I also have cultured tubes, and that was for a different experiment with different hormone concentrations. And I'm going to place one piece of surface sterilized tissue into each tube. You'll notice as I open the tube, I have been giving them a quick flame. I forgot to on that one. And I didn't do it on that one either. Oh well. So I completed that process for all the tubes, placed tissue in all the tubes. And finally, I took everything out of the laminar flow hood, and I put parafilm around the petri dishes to seal them. And this is one of those up close for the media it was in the petri dishes. This was the experiment where we want to induce callus from the carrot tissue, and it has IAA and kinetin in a 1-to-1 ratio, and for the concentration of each one is 0.005 milligrams per milliliter, or you could also say 5 micrograms per milliliter of each one. With this picture that's up close, you can see the outer layers of carrot tissue that were bleached out during the bleach treatment. And hopefully the inner tissues here are still viable and will turn into callus. And I prepared 11 different plates. Why 11? That was just because that's how much tissue I had. This was the setup for the second experiment for our class. In this one, we have 16 different hormone concentrations that were being tested. Each hormone concentration is in a set of 4 tubes, and each tube has a single bit of tissue. And with this one, we have concentrations ranging, starting with 0. There's no hormones whatsoever. And then we have different amounts of cytokinin and auxin in each one, and we're going to see what happens. With this experiment, it's more typically started with callus, but we started this experiment late this year, and or in this semester, and so I put whole tissue on there just to see what would happen. The results are going to be recorded for this experiment weekly and will be posted as part 3.